Okay, we're going to continue with muscles. This is lecture 7B. Now, typically, muscles in the body are maintained in a slightly contracted state. And that contraction is not to produce any active moment, but helps to keep the muscles firm and healthy. Good muscle tone, in other words. And this is due to spontaneous reflexes that occur. These are spinally generated reflexes that are constantly activating one motor unit and then another. And what they're responding to are just small shifts that occur for example, when you're trying to stand still. Truth of the matter is that your muscles are constantly contracting and relaxing in order to maintain you in that standing still position. So any wavering back movement, if you start to lean backwards unintentionally, then you have these reflexes that'll pull you back up. So you're constantly activating all over your body these skeletal muscles to maintain good tone and to maintain proper position or posture. All due to feedback from sensory neurons that act as stretch receptors. So you have sensory stretch receptor neurons, stretch receptor sensory neurons in your muscles and in your tendons that send feedback into the spine and trigger reflexes back out to relax or contract different muscles. Now, and it's a little hard not to explain it without it's easier to demonstrate this, but I'll explain it. There are two types of muscle contractions. One is isotonic, and in that case the muscle length is changed, it's shortened during the contraction. So you have a change in muscle length, and the joint across which that muscle is attached moves. It changes. The lower part across the joint is moving, so you're moving some kind of load. In contrast, the other kind of muscle contraction is called isometric, and that's just increasing the tension or force of the muscle. It shortens, it contracts, but there is no shortening of the muscle, so there's no movement of the framework of the body. The skeleton does not move at all. And you do this, without moving, just tense muscles in your body. You can tense your bicep without actually moving your lower arm. That's an isometric contraction, and this is the kind of thing that they recommend people do when they're on a long flight. If they don't stand, they recommend best to walk around every once in a while, but if you can't do that, then just do some isometric exercise, some isometric contraction of the muscles. And this is just they recommend that because it helps move the blood along so you don't get stasis of the venous blood and increased chance of getting a thrombus formation. So the other thing that I should explain to you about contractions is when we look at isotonic contractions where there's movement. So say, for example, just like we did in the lab where you had your arm was on the lower arm was stretched out on the bench, and you're holding a weight underhand in your arm, and you move that, you contract your biceps, the muscle in the upper arm, you contract that and move the weight up. Okay, so you're bringing the weight up to your shoulder, and then you bring it back down onto the table. You have a weight in your hand, you bring it up to your shoulder, and then you put it back down on the table. When the muscle shortens and you lift that weight and are bringing the weight up towards your shoulder from, you have your arm at say a 90 or 120 degree angle stretched out in front of you on the desktop, and you have a weight in your hand, you're holding it underhand, and you contract the muscle and lift it and bring the weight up to your shoulder, that's a concentric contraction. And then when you lower it down, you're still contracting the muscle, but you're allowing it to lengthen gradually in control. So you're generating some force, but not actually enough force to lift it up. In fact, just the right amount of force to let it down. And that's an eccentric contraction. So concentric and eccentric contractions. And here you see a setup in the lab where the muscle is isolated and it's attached by a hook to at one end, and it's attached to not a force transducer but a weight, and it shortens. So that's the equivalent to a isotonic concentric contraction, and the resting length changes and tensions develop. Okay, when we look at isometric contractions, the tension increases up to the muscle's capacity. Okay, the muscle is tensing, but it doesn't shorten or lengthen. It just stays at the fixed length, but the tension is increased in the muscle. And that's what's shown here. So there is a change in tension developing, and then you can relax after, but there's no change in the resting length. Now, we talked before about the sarcomere. Those were that's the sarcomere is the smallest functional unit at the molecular level within the muscle. It's the area on a myofibril between two Z lines. And this is where you have actin and myosin filaments arranged in an overlapping pattern, partially overlapping, and then when they contract, they overlap even more. So the sarcomere is the basic contractile unit within the muscle. It has a very fixed, repetitive arrangement of filaments, thin and thick filaments, so that the muscle is striated or striped in appearance. In most areas of muscle, if you look, you'll find that the myosin filament, there's a word missing right there, the myosin, for each myosin heavy, for every myosin thick filament, it's going to be surrounded by 6 actin thin filaments. Okay, myosin thick filaments and actin thin filaments. On this slide, what you see is a diagram showing the entire muscle. And this is the whole muscle here. And inside of that are these bundles of muscle fibers. These are called fascicles. Each one is a fascicle, as I've told you before. And inside that fascicle are several skeletal muscle fibers. And here's a single one pulled out. So this is one fascicle right there. There's a skeletal muscle fiber, and you can see on the edge here these individual separate nuclei. So together this is a multinucleated, striped or striated skeletal muscle fiber. And when you look inside, it contains several myofibrils. And if you look under the microscope at a myofibril, what you see is this very repetitive striped pattern, dark stripes, light stripes. This is a electron micrograph. These two diagrams are electron micrographs here. These electron micrographs or photographs from an electron microscope. And what you see here is a dark band, and on either side a light band. In the middle of the light band you have this dark stripe, and that dark stripe is the Z line or Z disc. This is the I band and an I band, and this is the, I believe, H band or H zone. Now when the muscle contracts, what you have is that the area where there are thick filaments, which is in here, slides past the thin filaments, which are here and here. And so the area that had thin filaments disappears essentially. They're sliding over each other, and now the Z discs, it's shortened, and the Z discs or Z lines are in those two positions, here and here. Those are the Z discs, right there, right there. Did I get it right? A band, yeah, that's the A band. Oh, I'm sorry. A band is the whole thing from Z line to Z line. I band is the light area on either side. A band, I'm sorry, I had it right. A band is the area where you only have thick filaments in there. And the I bands are on either side of the Z line. So you have a Z line there, right down the middle. Let me change the color of this. The Z line is here. This Z line is this structure here. And there's another Z line. The sarcomere stretches from Z line to Z line. The light area is the I band. So there's an I band across here, and there's an I band there. And then the dark band is the dark part stripe is the A band. The A band is right in there. So that's a single sarcomere. The thin filaments are drawn here. It's further expanded here. You see thin filaments, thin filament, thin filament, and these are the thick filaments here. So these are the myosin thick filaments with myosin heads projecting up that can bind to the thin filaments. This is the Z line and this is the Z line. So it's an overlapping pattern where you have both thin and thick, it's darker, and where it's only thin filaments, it's lighter. So it gives a very repetitive striped appearance. This is a diagram showing the similar thing, showing the sarcomere with the two Z lines or Z discs and the central A band and the I band and I band here. These areas are I bands, and the center dark part is the A band. This is in the relaxed state. That's when you can see all of these structures. When it contracts, then the I bands get narrower, thinner. So these are able to slide past each other during contraction. Here again you see the same arrangement with thin filaments made of actin, the protein actin, and the thick filaments here. Thick filament is there and there and there. And you get sliding past each other when you get contraction. I don't know if, yes, it's shown here. This protein is called, there is a protein here called titin. And these are elastic titin filaments. Titin is the largest protein, the largest known protein in the body. It's a really massive, huge size protein. So the A band is made completely, is an area of the myosin thick filaments. The I band is an area with no myosin, just thin filaments. And also you have that M line right down the middle. Here we go. This is a summary of that. And there you see the sarcomere with the two I bands and the light I bands on either side of the Z line. There's the, they're showing the A band slightly different, huh? The A band is the dark, thick filament area, and the I bands are on either side of that. There it is, the A band. And these are the I bands, right here and here, with the Z line, and the M band or M line is right down the middle. Thin filaments are actin, thick filaments are made of myosin. Again, the same thing, and this is a sequence of electron micrographs that shows what happens with contraction, I believe. So with contraction, what you're getting is attachment of myosin heads to the actin thin filament and then ratcheting along, moving it, they slide past each other. So here you see this kind of contraction occurring, and in this series of slides it gets narrow, the eye bands essentially disappear, because what happened was you get the thin filament sliding along, telescoping together with the thick filament. Okay, the mechanism of this involves binding of these myosin heads, these globular myosin heads that stick up from the thick filaments. They're all over the thick filaments, myosin heads. Each myosin molecule is a polymer of two long polypeptide chains coiled around each other. I don't know what it says under here. I don't think it's that critical. I accidentally covered it. Okay. This mechanism of muscle contraction is called the sliding filament mechanism. You have the thin filaments are made of actin. It's a helix of two long chains of multiple polymers, many monomers of actin that together form these two polymer chains. And twisted around that is a long filamentous structure, which is tropomyosin. All along here, I'm highlighting it in yellow here, that's tropomyosin. And at regular intervals, you have a structure, a protein structure called the troponin complex. Now, the troponin complex is essentially sequestering or hiding the binding sites for myosin. Okay, the myosin binding sites are on the actin filament, but they are covered by tropomyosin, that long strand of tropomyosin here. Okay, and what happens is that when an action potential arrives and depolarizes the skeletal muscle fiber and calcium gets released from the sarcoplasmic reticulum, the calcium will bind to the troponin complex, and that causes the troponin, and that causes the tropomyosin to change in shape and pull away and reveal the binding sites, the myosin binding sites that the tropomyosin covers. Okay, so on that thin filament, you not only have those two long chains made up of actin monomers and these two polymeric chains, but you also have that long structure of tropomyosin that's hiding or covering the myosin binding sites. And the troponin complex is what calcium interacts with, causing that interaction causes a shape change, causing a rotation away of the tropomyosin, revealing the myosin binding sites. Okay, so with that movement away of the tropomyosin and exposure of the myosin binding site, you get myosin heads can now bind to the actin, and the myosin heads have ATPase activity. In other words, they can cleave ATP, releasing energy. So with binding of the myosin head, there's changing of the shape, and the thick and thin filaments move past each other. You get contraction. And then, so they're sliding closer, they're overlapping more. And the myosin head then will bind ATP, and and with that, you get release from the myosin. So with energy being released, the myosin molecule detaches from the myosin head, detaches from the actin, and then is able to again bind if calcium is there and the myosin binding site is revealed, then myosin can again bind to the actin and the two strands can slide along each other. But it's the release away of the myosin head from the actin that's the energy dependent step. So because in fact that the energy, the ATP, is required for release of myosin head from actin, and because when a person dies they are no longer producing any ATP, then soon after death muscles start to stiffen, and this is called rigor mortis. This is the rigidity or stiffness of death. So the body becomes really all the muscles are quite rigid. That's why in slang, some people call a dead body a stiff. So because there's no release of the myosin from the actin, the muscles stay in a contracted state, and without ATP they don't release. They stay that way for a long time, but for quite a few hours, but eventually it does release on its own. Okay, so this is just for your own interest, that these proteins actually have structure made up of many amino acids strung together in quite a complex three-dimensional shape, and it is the three-dimensional conformation that gives the molecule its function. So here you see the myosin head drawn by an artist, and here you see it as a space-filling model with different areas, subunits or domains colored differently. But these are many amino acids are shown here, and there are different sites on that. There's a ATP binding site where it binds ATP before the ATP gets hydrolyzed to release energy. There's an actin binding site here, very specific. That's where the myosin head binds to actin. That's just for your own information. Okay, so let's just go through it. The events associated with release of calcium from the sarcoplasmic reticulum following excitation. So you have the arriving action potential along the axon, releasing acetylcholine at the neuromuscular junction, and the depolarization of the skeletal muscle fiber membrane, surface membrane, the sarcolemma, and that depolarization spreading, or action potential spreading down the T-tubules, triggering calcium release from the sarcoplasmic reticulum. Here you see a thin filament actin filament, and the tropomyosin is hiding the myosin binding sites. The troponin complex is there, and if calcium binds to it, then you will get exposure of the myosin binding site on the actin filament, so that the myosin head can now bind to actin. So let's see what happens here. That white sphere, I guess, is the calcium, and you see along here the myosin binding sites are being revealed, those yellow spheres, and now the myosin head can bind, and you get ratcheting movement along of the strands, overlapping more. That's what these arrows are suggesting. That's shortening of the muscle because the thick and thin filaments are running past each other, overlapping. ATP then is binding to the myosin head and is hydrolyzed to release ADP and phosphate and some energy, because with the release of that phosphate you get energy release, and this allows for the myosin head here to release from the actin filament, and then it's ready again to bind again if there's still calcium, if there's still ATP in the muscle. Okay, let's move on to smooth muscle. We're going to actually go through smooth muscle very quickly and not say that much about it. It's quite different from skeletal muscle. We've already talked about cardiac muscle, so when we did cardiovascular physiology. So here we've spent a lot of time on skeletal, but we'll spend a little bit of time on smooth muscle. It is very simple structurally. You have a single cell. I'm going to see if I can draw something that resembles a single cell. So you have a single long sort of spindle-shaped cell with a single nucleus, and these are arranged in large sheets running, cells running parallel to each other. You can see up here in the diagram. No, I didn't want to do that. It's a little difficult to see there it is. Okay. So what you have are these long cells all lined up parallel to each other. I'm just highlighting the nuclei, but you can see that the nuclei and then therefore this long cell, they're all parallel to each other. They typically form these sheet-like structures of parallel cells that are all arranged in a sheet. Smooth muscle is typically involved in contraction around visceral tissue and is present in tubular structures in the body, like the digestive tract, the vasculature, the respiratory airways. As I said, they're arranged in sheets, and the smooth muscles are coupled to each other electrically by gap junctions, so the whole sheet can contract together. It's not a multi-nuke, it's certainly separate cells, but they are electrically coupled through those gap junctions, so action potentials can spread quickly through the sheet. Now, smooth muscle can contract, but it's not under voluntary control like skeletal muscle. It's controlled by the autonomic nervous system, so it's involuntary tissue. It gets activated by the autonomic nervous system, either the sympathetic or parasympathetic division. So these stimulation of by neurotransmitter will alter membrane potential and allow for contractions or relaxation of smooth muscle. And it really depends on where in the body. It all depends on what kind of receptors, neurotransmitter receptors they have, whether you get more or less contraction of smooth muscles. So different areas of the body will respond to different neurotransmitters released by the autonomic nervous system innervation in different ways. The other way that smooth muscle is controlled is a built-in intrinsic response to stretching. Smooth muscle can stretch. It can be passively stretched, and with that, typically what happens is that you get depolarization of the tissue, firing of action potential and contraction. An example of that would be with a bolus of food moving through the digested material moving through the intestinal tract, stretching the smooth muscle in the wall of the gastrointestinal tract and triggering contractions. Those contractions of the wall of the digestive tract or GI tract will allow for more mixing of the material, mixing with digestive enzymes, and also push it along and move it forward. Now the other type of muscle, and we've done this in cardiovascular physiology when we talked about the heart, the other third kind of muscle is cardiac muscle tissue. These cells are striped, although it's not that easy to see it in this photomicrograph, but there are striations. Again, because of the regular arrangement of the thick and thin filaments made of actin myosin. Thin and thick filaments made of actin myosin. They are branching cells. If you have a cardiac muscle cell, let's say that's the nucleus, then the cell will be, stretched out, multiple branches stretching away from the cell. And at the ends of these branches, what you have is a unique structure which is present where the next cell branch is, and it's right there at the ends, and these are called the intercalated discs. So you have intercalated discs at the ends of the branches, and they act as a they act as a joint from one cell to the next, and allow for conductivity of signals because they have multiple gap junctions within them. So overall, the cells that are all attached here in the cardiac muscle tissue, they're all attached through these intercalated discs, can act together as singles, what's called a syncytium. So they can act together, and the whole area of cardiac muscle can contract as one. So depolarization at one point in the heart, which, depolarizations occur spontaneously with pacemaker tissue in the sinoatrial node, will spread not only down to the AV node, the atrioventricular node, but also right across all the atria through all of these cardiac muscle cells and triggering them all to contract as one. Remember that the pacemaker cells are in fact actually specialized non-contractile muscle cells that have automaticity. They spontaneously depolarize. They don't need any outside signals. The pacemaker depolarization can be modified in terms of the rate, slowed down or sped up, but they're not dependent upon signals from the autonomic nervous system. The autonomic nervous system, sympathetic division of the autonomic nervous system, will cause an increasing rate of depolarization, therefore speeding up of pacemaker depolarization and therefore speeding up of the heart. And parasympathetic will results in a slowing down, but the fact that they'll actually depolarize, the heart will depolarize and beat because of the pacemaker, does not need attachment of the or any influence by the autonomic nervous system, and that's why heart transplants are possible, because of that built-in pacemaker activity. Okay, I believe that's the last slide, and now I'll go back to see if things are okay with the recording.